Okay, now live telecast has been started to YouTube and Facebook. Good afternoon, everybody. Good afternoon, everybody. Good evening. Now we are going to start our workshop. Workshop with two important speakers, Dr. Dilip Gore and Dr. Devdatta Nayak. I request Dr. Gajbe, Jyoti Gajbe from the Department of Community Medicine of this institution to give their introduction. Dr. Jyoti Gajbe, please.
Yes, sir. Sir, am I audible properly?
Yes, yes, yes,
Hello?
yes.
Yes, sir. Okay. Thank you, sir. Good afternoon. Good afternoon, our respected to our lovable director, Dr. M.G. Zite, sir, Principal, sir, Dr. V.C. Gore, Professor Dr. Akhilesh Khan, sir, our Superintendent, Dr. Bhabat, sir, Dr. Karthik CEO Bahula, and good afternoon to all of my colleagues and my dear students. A very warm welcome to all of you. Myself, Dr. Jyoti Gajbhiye, a part of the Antarbharti Homeopathic Medical College and Hospital, Dabha, Nagpur. I am working as an assistant professor in Community Medicine Department. It's my pleasure that today I get opportunity to introduce such a researchers who are beyond the description, and today they are with us as a speaker. So let us introduce to our today's speaker who are having such a long journey in the field of the research. Our today's speakers are, the first is Dr. Dilip Gore sir, who is PhD in biotechnology, and second is Dr. Devadatta Nayak sir, who is MD in homeopathy. So let us introduce our first speaker, Dr. Dilip G. Gore sir. Uh, Today he will talk on the topic role of nanotechnology, drug carriers, and molecular approach for the better health. If we say about the qualification of Dr. Gore sir, he is PhD in biotechnology and molecular entomology from the Nagpur University. If we say about profession, he is acting as a founder director of Sai Biosystem Private Limited Nagpur since 2013 till date. He is also acting as a director of MacDill India Private Limited Nagpur since 2017. His specialization is molecular biology, bioinformatics, comparative genomics and proteomics, biopolymers, hydrogel, and nanotechnology drugs. He is on the panel as an advisory member of Dabha Homeopathic College, Nagpur, advisory committee member of Nagpur University of Biotechnology subject. He is advisory member of IP cell at the Dhanvade National College. He is also the member of examination section for Yashwantrao Naik, Yashwantrao Chauhan Maharashtra Open University, Nashik. He is also as a reviewer for he is also as a reviewer of African Journal of Biotechnology, elsewhere Journal of Pharmacy Research, Indian Journal of Microbiology, uh, Spring Publishing House. He is the guide. He is guide of the three PhD students for biotechnology, MIT University Raipur under collaboration. He has delivered about 80 guest lectures. He published his 37 research articles, that is international and national level articles related to the molecular diagnosis, vaccinology, functional genomics, virology, and antibacterial therapy. Dr. Gore sir also formulates some products. They are recently developed. The recently developed products are biodegradable plastics, which can be used in PPE kits and other commercial usage. He developed hydrogel, which can be useful in ophthalmic lenses, and he is heavy metal and is in heavy metal absorption in industrial use also. He formulate these two products. So this all about our first speaker, Dr. Dilip Gore sir. Thank you, sir.
Now I request Dr. Gore sir, Dr. Dilip Gore to start his program. Dr. Dilip Gore sir, please,
Yeah.
if you need skin sharing, you go through the skin sharing.
Is it visible? The screen is visible?
That's right. Now visible.
Okay, okay. So very good afternoon to all of you. First of all, I would like to thank the Antarbharati Homeopathic College and its management, along with the Bahula Foundation, for giving me an opportunity to talk in front of all the eminent personalities who are doctor in profession. Uh, Today, I will be talking about the nanotechnology and the role of homeopathy, how the nanotechnology is actually playing a very important role in homeopathy. This is all what we are talking in the next 1 hour of time. So I think PPT is visible to all of you, and I'm also visible to all of you. Is it okay?
Yes, yes.
Okay. So my topic of presentation today is of a role of nanotechnology, which could be a drug carrier, and what is the exact role of these nanotechnology molecules into the homeopathy on a molecular level, and by which how we can get the proper or better health by the treatment or by the doses of the homeopathic medicines. This is what we are talking about in this lecture. So, in an introductory part, we'll talk about the homeopathic medicines. Since we all know that homeopathic is a very old science, actually, who is more than 200 years of the having the history of working. It is now classified as a complementary and alternative medicine, as we all know. But there are some skepticism always followed by this therapy. But still, it remains the mainstream of medicine, and now approximately 500 million people worldwide accepted this therapy for the better health. The main reason for the acceptance of this therapy is its low risk treatment approach, which can be applicable for the acute and chronic conditions also. Now, in today's era of research, where we have a number of facilities of clinical and health services and instrumentation, homeopathic has now come up with the strong evidences of their physiochemical properties, their biological activities in vitro and in vivo. And now this is giving us the real world effectiveness for the benefit of the society. But homeopathy never remained beyond skepticism. So skepticism kept on repeating or returning to on the belief. Means many people say that homeopathic remedies per se cannot be exert any effect other than a placebo effect. Means they are saying that most of the time homeopathic is nothing but the placebo. But what is the main reason for this is that homeopathic is claimed to be implausible, means non-effective on a scientific ground when we are looking at the formulations. So skepticism always reveals that okay, what homeopathic is doing, they are doing serial dilution component of the remedy manufacturing. Means whatever the component, whatever the material you are using as a bulk form, you are diluting it, and you are diluting at such a level that it is not becoming an effective. This is always the skepticism kept on rolling with the homeopathic part. But always the skepticism ignores the some of the basic science and the preclinical data which homeopathic has showcased in the recent times. They also discount the many positive clinical studies. And that's why we have to find out the solution or the remedy towards the skepticism. So with this skepticism and uncertainty about the nature of the remedies and their mode of action, how this science can grow. But because of this, it remains slightly slow spread acceptance medicine. And now it has not become an integrative medicine till date. So to solve this problem of skepticism, there is a chance for the researchers to answer the skeptics that how homeopathic research is useful and how homeopathic research is going on to prove the effectiveness of the drugs or the medicines which we are utilizing. So till date, thousands of homeopathic patients suggest that yes, they have seen improvement in themselves. About 60 to 90% patient always says that okay, we are working fine, we are getting a better health because of the treatment of the homeopathic as compared to the previous drugs what we have taken. So this is one of the success story of the homeopathy. Now, not only is that, what is the real requirement in the homeopathic part is that what is the prerequisite? The prerequisite is that what homeopathic researchers can do. Now, this is very important part in today's life to prove how and what is the exact mechanism homeopathic is working on. So, if a researcher can give a clear understanding of the homeopathic remedies, and how they are actually working in their mode of action. This will help the researchers, this will help the clinicians to put up a strong literature, and based on that they can go for the better side of the they can put up the better side of the drugs. So what will be the ultimate outcome? The ultimate outcome will be that homeopathic clinicians will be able to practice clinically relevant evidence-based care. Now this is important, clinically evidence-based care. They will come to know what is the exact mode of action of the drug and how it will be given. So taken together, the available scientific evidence now points to a testable comprehensive model for the nature of homeopathic remedies. So what is required is the testable comprehensive model for the nature of the homeopathic remedies, how they will act, and what will be their exact interaction with the living system. This is a very important aspect now comes up in front of the
Any problem? Any problem? Dilip Gore is not audible to us. Hello? No sound. Yes, wait. Dilip Gore has left. Probably network issue is there.
I think there must be a network
I'm contacting.
difficulty.
What is the problem? It is showing connecting, but our host is telling it is network issue. You are not connecting, not connecting. Not connected. You are not connected. Network issue is there. So please just he's trying to connect. Wait, he's connecting with us, trying to connect because there is a network issue. Dr. Dilip Gore has joined. Please open your audio and video, Dr. Gore. Dr. Dilip Gore, please open your audio and video. Dr. Dilip Gore, you have been connected now. Again left. No, connected, now connected. Please unmute your audio and video. Please start your video. Hello. Yes, Dr. Karthik, please allow him. Dr. Karthik.
I've already done that. He's already a co-host.
And Dr. Gore, please open your audio and video. Dr. Dilip Gore, please audio and video. Open your audio and video. What happened with you? Again left. There is a severe network issue. Hello. Hello. You left again, you joined again, left. Hello. Your network is, I think, very disturbing. And now again join. You please rejoin. You please open your audio and video. You have been allowed.
Yeah.
Now you please start your program.
Yeah.
Okay.
Sorry for the disturbance.
It is not in your hand.
Yes. So we were talking about how the remedies, how the homeopathic drugs are synthesized. This is very important to understand. Now, as you can see here, that homeopathic drugs are actually prepared by two important process. One is known as trituration, that's what you can see here. And what is known as trituration, which is a mechanical grinding. Now, what is this mechanical grinding? Here, what they are doing, they are actually whatever the drug base material, or what is a source material, what we are using for the synthesis of the drug, they are simply grinding it in a mechanical process. So whenever we are grinding any content under a very high pressure, the big size or the large size molecule are getting converted into a very small size molecule. And if the continuation of the grinding takes place, these small molecules will get converted into nano size material. Now, this is very important in terms of synthesis of the homeopathic remedies. So this is the step actually helping us to form the nanoparticles in the homeopathic drugs. After getting the mechanical grinding of the molecules, we are going for the succession. Succession is about nothing but the intense agitation of the chemical material or drug material, which is a source material of the homeopathic drug, under an ethanolic solution. We are continuously agitating the content or material under the ethanolic solution. So these two processes actually forming the nano size material into the homeopathic remedies. And this is the very important part. Now, whenever this process is taking place, we are doing this in some kind of a material, some kind of a container, okay? And for the most of the time, we are using a glass material for the synthesis of homeopathic material, drugs. And glass, as we know, it is nothing but the silica, okay? And now in some cases, we are also using some polymers like polyethylene. Okay, so these containers of polyethylene or a silica also helping us to form the nanoparticles. Okay, that I will discuss in the coming time. So ultimately what happens during the homeopathic remediation formation, we are getting the number of nanoparticle formation. Now, these small nanoparticles, we are saying that they are the actual active ingredients in the homeopathic medicines. Now, does it mean that homeopathy is based on the nanoparticle therapy? This is what is the question comes in front of us. So we have to give an answer for this. So yes, I can say homeopathic medicines or the remedies does contain the number of crude nanoparticles. They are nothing but the nano aggregates or the nanocrystals of their original source substance. Original source substance means it may be a plant source, animal source, mineral source, something like that. So this is a very important term. It's a nanocrystal or a nanomaterial of original source substance. So first thing we should keep in mind is that homeopathic medicine contains the nanoparticles of original source substances. This is one of the component of effective ingredient. So homeopathic remedies, now we can say these are the nanoparticle forms available from their source material and because of which they are now formed. Now, remedy manufacturing begins with the mechanical grinding, and that what we have already seen. Now we know the grinding is on mainly takes place in a lactose medium. Lactose is one of the sugar source in which the grinding takes place. So vigorous agitation in the ethanolic water solution or in a lactose gives us the nanoparticles. Now, very important point to be noted. Whenever we are synthesizing a homeopathic medicine, what is important to understand is that in the history or till now, we are synthesizing homeopathic medicines into the glass containers. But now some of the manufacturers are using polymer containers like of polypropylene and polyethylene. So it is very important to see that if we are synthesizing such drugs into polypropylene and polyethylene, possibility is that nanoparticles of polypropylene and polyethylene may come into the formulations of the homeopathy. So this is very important. So this what we have seen up till now, that synthesis of homeopathic drugs gives us a very important source of nanoparticles. Now, during this process, there is one important step comes up that is known as succession. Now, succession is nothing but the a fluid turbulence. As you can see into this image, this guy is doing the same thing. He is doing the fluid turbulence during the process of succession. And succession, what is happening here? There is a continuous particle collision. And because of this continuous particle collision, it shears off the smaller particle into a smallest particle, and ultimately they gives a formation of nanobubbles. So now this is very interesting that while doing the succession, there is a formation of nanobubbles. Now what are these nanobubbles? These are nothing but the tiny areas in the localized high with a localized high temperature and pressure, which has the ability to give a smaller nanoparticles. Okay, and ultimately it also captures some of the silicate nanoparticles because we are doing this reaction into a glass material, which is nothing but made up of silica. So this is very important, that succession is also giving us the nanomaterial in our drugs. Now, what is the nanoparticles of homeopathy? Now, this is interesting part, that if I say homeopathic is nothing but the treatment through nanoparticles, what are they and how they are forming and how they are acting? These are very important. So we have already seen how they are forming. Now question comes, how they are acting. Okay, so first we should know what is a nanoparticle is. Nanoparticle is nothing but that it's any source material whose one dimension should measure less than 100 nanometers. So out of the three dimension, one dimension should be less than 100 nanometer. Okay, most of the time we are synthesizing the drugs of homeopathic in ethanol which are giving us the smaller size nanostructures. Okay. Now, if it is so, then we are saying that but most of the researchers are telling is that nanoparticles are toxic to the human body. Okay, this is very important. So if it is so, how homeopathic is treatment is sustaining such a treatment? Am I audible? Voice is clear to all?
You are audible, but your voice is not clear. I think there is a network issue, severe network issue. Your voice is not clear. Are you audible to you? Are you audible? Are you audible to me? Are you audible? Are you audible to you? Again, disconnected. Dr. Kartik, Dilip Gore is connected, not but severe network issue. What is the problem? Your network, severe problem there.
Now it is clear, coming to you. PPT is coming to you.
Now clear, now clear.
PPT is clear coming to you.
Not PPT is not coming.
1 minute.
Now wait. Yes.
Now it is coming. Hello. Hello.
Yes. Audible.
Okay. Now, so these are the very important points, that if the homeopathic drugs are containing nanoparticles, whether they are toxic to us or not, it is very important. Thus, nanoparticles are the things which we need to keep in mind. First thing is that if we use a smaller size nanoparticles, they are always tends to be toxic. Okay, if we are using the smaller size, they are toxic. Okay, not only that, if the drug is containing nanosilica and nanopolyesters, nanoparticles, they are still proving to be toxic. So this point we need to be considered. Now, another thing is that nanoparticles, on the other hand, giving us the bioavailability, means they are very effective at a very low concentration as compared to the bulk form. Now, if we are using a bulk form, means maybe an allopathic drug which we are using in a bulk form, maybe in mg or μg, which is a bulk form of it. But whenever we are using any homeopathic medicine, it comes in a very low concentration. And this is the very important part, because they contains nanoparticles, they are very low in concentration and yet they are effective. So what's that means? So that's why homeopaths utilizes low doses instead of giving them at the optimum or up to neutrality. This is a very good example that homeopaths are never giving the drugs to the optimum or up to neutrality, and still the drug acts perfectly. So this is the interesting part what we have to understand. So ultimately, if you have to minimize the adverse effect of any drug, it have to give it at the lowest level. So nanoparticles of homeopaths are doing the same thing. It is giving us the beneficial endogenous adaptive response in the form of the nanomedicine dosage. And that's why they need in a very low quantity. So how homeopathic nanoparticles are formed? As I already told you that nanoparticles are formed by grinding and by succession process. Along with that, they are forming the other source nanoparticles, which I already told you, that is a lactose nanoparticle and silica nanoparticles. Lactose, which is in use during the process of synthesis of drugs, and silica, which is a glass material, or the instrument, or the what you can say, a vessel in which we are forming the drugs. So these two molecules of lactose nanoparticles and silica nanoparticles, they always remain as nanocontaminants in the drug of homeopathic remedies. So they are the nanocontaminants in each and every homeopathic remedies which are available, if you are synthesizing them in the presence of lactose or in the presence of glass. But what are their real role? They are actually acting like a nano adjuvants. Now, what is a nano adjuvant? They actually brings about the many reactions inside of the body once they come in contact with the cellular system, and they help in a drug delivery vehicles. So they are actually the drug delivery vehicles, and that's why they are also useful in reducing the dose level. So the use of lactose and silica nanoparticles is very crucial in the homeopathic remedies. So, but now what is happening is that many times people are using polyethylene instead of the glass tube, and that suggests that instead of the silica nanoparticles, we are getting the polyethylene nanoparticles. Now, this we have to look after that. What is the real effect of such kind of a nanoparticles into the homeopathic remedies? Now I have told you a number of things about this. Okay, this is what is the recent findings about the homeopaths and all. But the great Samuel Hahnemann, uh, in his finding, put forward this record actually, but he was not clear at that time because of the unavailability of the techniques and the methodologies which we are having right now. Now this is the what is the main finding what Samuel Hahnemann has given during the process of formation of homeopathic medicine. He told us that there are some molecules which are forming and which are nanoparticles, but he hasn't given the name nanoparticles at that time, but he mentioned it. Now what he has mentioned it is very important. Now what he is saying, you can see in this second statement, he emphasized that the role of trituration requires the prolonged intense mechanical grinding of the source material lactose with mortar and pestle. Means they said that your source material must undergo a trituration for a prolonged intense grinding in presence of a lactose having the mortar and pestle as a supporting material. Then you should go for the succession, so that the and that too in a liquid container, liquid material having a glass container. And if you do these two things, means trituration and succession, then and then only you will be able to generate a very important active homeopathic remedies. Now this is very important. He said in his findings that if you go for the trituration and succession, then and then only you will find the active homeopathic remedies. Now this is a very important point. Now, the support has been given in this in his book also, that Organon of Medicine 6th edition, which has been completed in 1842. Now, what he said, this remarkable alteration in the property of the natural body is achieved through mechanical action. Means if you use only a natural material, and if you put into some liquid and if you dilute it, you will not get any good material. But if you do the trituration and succession, these particles are separating from one another by intervening in different substances, and ultimately you will get a undisclosed power that is discloses this power more and more. Means if you grind it, you will get more and more properly formed nanoparticles. But he has not said like that. But now we can clearly indicate it. We are clearly showcase that grinding and this is actually giving us the nanoparticles. Now, still the skepticism kept on coming towards the homeopaths. What skepticism is that? Skeptics always says that homeopaths are doing the serial dilution of the remedy material, and if you are diluting it so much potential, means 10 parts 100 times dilution, how you are claiming that this simple plain water is effective? Okay, and this claim of the skeptics also been agreed by the Henman in the earlier part, but he said that you are correct, you all skeptics are correct, but only with the bulk form. But what we are trying to do is that while forming the homeopathic drugs, we are diluting it, but not without the trituration and succession. Means if you do the formulation and the dilution without trituration and succession, it is definitely a placebo. It will not gonna work. But if we do the trituration and succession and then do the dilution, we are getting a real remedy. This is the what is the answer given by the Henman at that time. And this has given the strong blow to the theory of the skeptics. You are correct. If you do the dilution without trituration and succession, it is nothing but the placebo. It is not going to give you any good effect. But if you do the trituration and succession, the material in the nanoparticle formed by the trituration and succession will give you the nanomaterial. He has not said like that, but he said that something is happening, something miraculous is happening, something powerful is happening, and that gives us the a drug. Now today we are having the number of methodologies, number of techniques to prove it, and one of the technique is a transmission electron microscopy, which onto that basis we can say nanoparticles are forming. So one of the Chickermani et al. Demonstrated that the formulations, the material formed or the homeopathic metal remedies formed by the 6 different mineral-based solutions are having the presence of nanoparticles even at the liquid potency of 6C, 30C, and even at the 200C. Means even we are doing the 200 times dilution, we can see the presence of nanoparticles in that. So which confirms that bulk material, if you do dilution, it will show the lesser concentration, but there is no effect onto the concentration of the nanoparticles. And this is very interesting part in the homeopathic side, that the drugs are containing the nanoparticles even at the 200C potencies or the dilution. So, so this is what they are saying. But if you are not doing the trituration and the succession, it will definitely not going to give you any good remedy even at the 12C. So this is very important part to do in the homeopathic preparation. So skepticism kept on coming, and our faith is going on increasing. This is what is the term said by the Oscar Wilde. So now we have number of evidences already with the homeopathic drugs that yes, they contains the nanoparticles. The question comes that if we are using a bulk form of the material, if we are using any plant source or animal source or minerals, if I am not forming the nanoparticles out of it and just I am simply diluting it, why they are not so effective as compared to the nanoparticles formed from them? If you are forming the nanoparticles, then they are becoming effective. And if you are not using them in a nanoparticle form, they are not forming any effective results. Now, why this is so? This is the answer for this. Now we have utilized a number of advanced methodologies like Raman spectra, UV spectra, XRD, EDS, scanning electron microscopy, transmission electron microscopy, fluorescence spectroscopy, which has revealed that the homeopathic remedies are actually having the varied forms of nanoparticles. And they have the different features actually from the plain solvent controls, means only the solvent what we are considering. They're also showing that there are different types of nanoparticles are there which are differ in their thermal, electrical, and optical properties as compared to the bulk form. What does this mean? This means that nanoparticles, if they are forming into any remedy, they are changing the thermal, electrical, and optical properties of that component. And if they are not forming, it will remain the change. It will remain in the change form. It will remain in a different form. So this is very important to find out. Another thing is that homeopathic remedies also contains quantum macro entanglement like characters. And this is just because of the presence of the nanoparticle. So it is important to understand that if these are the changes occurring in the material, in the presence of the drug, in the presence of the nanoparticle, drug will act differently. So, resultant what is happening? There is an enhanced bioavailability and biological reactivity of the homeopathic remedies because of the presence of nanoparticles. And that, see, that is the reason we are using it in a very low dose and still it is giving us the good magnitude of results. Now, so that's why this is the final blow to the skeptics that, okay, we are using it in a very low dose, still it is performing. Why this is so? So this is very important. What is skeptics is saying, it may be correct that bulk form of material are serially diluted out of the solution past Avogadro's number. Means if you are diluting a material up to the past the Avogadro number, it will not be any more than a placebo. But study suggested that even you are doing the dilution past Avogadro number, you are not changing the nanoform source of the material. Means nanoform source will remain the same. They will not gonna change. They will not gonna alter. And this is the final skeptics blow given by the advanced methodologies of nanotechnology. And it helps the homeopathics to to explain how they are actually working in its mechanism, even at a very low concentration. Now, so what we have understood up till this is that, yes, drugs are containing, homeopathic remedies are containing nanoparticles. Now, question comes, whenever we are synthesizing them in a companies, in a manufacturing companies, how we will assure that the nanoparticles will remain of a same quality, how they will remain of a desired values and all. Now this is the real question remain in front of us. I already wanted to say you that different companies are utilizing different manufacturing processes, even though the protocol remains the same, but the process change. If something is changing, if we are by diverting something from the constant versus variable, the nanoparticle quality will and the quantity will change. And that's why the drug quality will also change as compared to each other whenever we are going for the different manufacturing company formulations. So data suggests that potential of or greater variability of nanoparticle quantities and perhaps the sizes and the shapes at a higher homeopathic potencies are currently manufactured. Means companies are producing nanoparticles which are available in homeopathic remedies, but they are varying in their quantities and the sizes. And this has been evidenced in the gelsemium remedy, which has been used for one of the drug formation. So it is important that this has to be tackled out whenever we are going for the synthesis of the drugs. Now, if this is the thing that quantity and the shape of the nanoparticle matters, how we can assure that the drugs will remain in good quality or a quantity. So it has been said that whenever the synthesis of any homeopathic remedy is going on, maybe from minerals, animals, or plant source, we have seen the surface irregularity. We have seen the surface irregularity in nanoparticles, but there is no major change in the size of the nanoparticle. So this is a good thing actually. We are getting a constant size nanoparticles. And because of that only, most of the time, homeopathic remedies produced by the different companies are giving us the better results. So it is important that not only the quantity, but also the size, shape, and features of small particles are very significant in giving out the biological effect. Hence, we have to go for the research towards this part, that how we can maintain the constant shape, constant size of the nanoparticles which are available in a homeopathic remedies, so that we can make it universal, we can make it constant for the remedy. Now, we have already said that nanoparticles are formed from the number of biological origin. plant source material, animal source material, but drugs of homeopaths also contains the silica nanoparticles. Now they are very important, silica nanoparticles. Now what is this silica nanoparticles? Whether they are present in every drug formulation or not. So if we are synthesizing homeopathic remedies in a glass container, I would say every time you will find the silica nanoparticles into that. Okay, and they are nothing but the nano contaminants. They are nano contaminants. They are not the material coming from the original plant source material. It is just coming from the container what we are using. Okay, so this is a very important part to be included. Now, how these nanoparticles, especially the silica nanoparticles, are working? Silica nanoparticles are highly reactive and adsorptive. They are very much reactive, and they can absorb number of bioactive molecules onto their surface. And whenever they goes along with your homeopathic remedy into the system of the body, human system, it augments the immune response. Okay, it augments the immunological response, and because of that, it acts like a signaling system to the immunological response. So no matter what kind of drug you are using, no matter what type of remedy you are using, but if you are preparing a drug in a silica container, you will find the presence of silica nanoparticles which are involved in immunological response. This finding has also been covered by the Samuel Hahnemann in his writing. What he said that patient, whenever they are using a homeopathic drug, it should be administered into the water, and you should go for the additional succession. You should go for the rigorous, what you can say, shaking of the material in a treatment bottle. And then only you should drink it, rather than simply putting a dry pellet into your mouth. He said like that, but he was not knowing what is the exact action. Now we can clearly indicate it. Now we can clearly showcase that he said that it is because of the silica nanoparticles which will go through the bottle in your body, and it will give an immunological response, and which will help you to give out a better response of your drug. So at that time, there was no such methodologies are available, but now we can say like this. So if you do the succession, it will release more and more nanosilica, and which will stimulate the immunological response. This is very important point. And nanoparticles now, hence especially the silica nanoparticles, are now acting like a vehicle for the other drug nanoparticles to carry out into a system for the response. Now, if I'm saying like this, that homeopath is nothing but the nanoparticle usage material, people are now going towards the nanoparticles. Everybody knows that success story of nanoparticle. But again, this remains a Star Wars concept. Most of them are in a research mode. They are not going in a practical mode. Everybody is doing a research on nanoparticles, how it will be effective, how it will be going into a response. But homeopath has already proven that nanoparticles are effective at a low dose concentration. So going through that, the same story, going through that same success stories, now we are using nanomaterials in a different form, which may be a nano drug, which may be a nano vaccine, which may be a nano gene delivery system. So this we know that nanoparticles are doing wonders if we are carrying out the studies in a controlled conditions. We are also getting now nano vaccines, which are giving out the immunological response at a very low dose. That is of only 2.5 nanograms will give the 1,000-fold higher immunological response. This is one of the indicator that if we use a nanomaterial, it will elicit the immunological response. This is the same story happening with the homeopathic medicine. So now the questions comes with the homeopathic remedies. We have learned, we have understood that, okay, homeopathic remedies are having nanoparticles. Then some questions comes with this. First thing is, if homeopathic remedies are nanoparticles and nanocrystals of the source material, how would they exert a therapeutic effect? Now, if they are a nanoparticle or a nanocrystals, how they are becoming a therapeutic molecules. Second question comes, aren't these nanoparticles are often toxic for the cells and organisms? It has been told many times that nanoparticles are very small molecules and they become toxic to the cells and organisms. Then homeopathic remedies must be the toxic material. But we have to answer this. Third question, even if the homeopathic nanoparticles are benign, means they are not at all affecting a cell. Benign means not affecting any cell. Then how could such a small quantity in a low dose used in a homeopathic remedy initiate a large organism-wide changes? If I'm saying they are benign, they are not affecting cells, then how they are affecting whole body system? This is the third question. And last, and very important, is that if we give sometimes a drug for a one particular disease, you will find some unrelated diseases also getting cured by the homeopathic remedies. Now, how this is happening? These are the four major questions comes in front of our mind whenever we are looking in terms of homeopathy and nanoparticles. Now, to these questions, we should have some answers. So these are the answers which we can see in a broader way. What is actually homeopathic remedies in the presence of nanoparticle are doing? First thing is they are going for the biological stress response. They elicit the biological stress response. Second thing is they allow a system to undergo a physiological adaptation. Third, they are going for the cross adaptation. And fourth is they can carry out cross sensitization. Now, what are these 4 points? What is a biological stress response? Biological stress response may be a system inside of our body where if any foreign material comes inside, maybe in a low dose, our body will consider it as a threat. It will consider as a life surviving opportunity that we have to work against it. So most of the time, whenever we are giving homeopathic remedies, body considered this as a stress. They are considering that this molecule is a stress, and they started to respond in a system, and that system they initiate is known as stress response system. And this is a whole body response. This is a whole body response which will act in together to control the stress. But while doing the stress controlling, they are also controlling the other pathogens or other foreign materials which are coming in way. And ultimately, it is a complex adaptive system of the homeopathic remedies, which is helping us to get cured whenever we are giving the dosage. Physiological adaptation is there, then cross adaptation is there. Cross adaptation means if you are taking a drug for one particular problem, it is also helping to get cured for the second problem. And this is what is the main advantage of using homeopathy. Now, this cross adaptation is actually occurring because homeopathic drug, we are not saying that it is a very much target specific. It is because it is not a target specific, it is a system specific. It is helping a whole system to work in together. And that is what is the main advantage of using homeopathic remedies. And cross sensitization means whenever we are going for the one organ treatment, it will also give the response to the another organ, and they will try to find out if there is a complexities in our body, we can cure it out. Now, this is a very important answers given by the homeopathic remedies, and in these 4 ways the drug functions. Now let's understand how the homeopathic drug actually works. This is very important. If I'm saying homeopathic drug is nothing but the nanoparticles, then action of these 2 molecules, maybe homeopathic remedies and maybe nanoparticle, they should work alike. They should work like same. And these are the answers given. Yes, both nanoparticle and homeopathic remedies actually can initiate hormesis. Hormesis, very important word. Yes, homeopathic remedies can initiate hormesis. Now, what is this term hormesis means? In the term pharmacology, toxicology, and physiology, the hormesis refers to the universal biological adaptive process. It's a universal biological adaptive process of a living organism in response to a low level of environmental stressors or toxins. Homeopathic drug is a very low environmental stressor or a toxin against which our body responds in a universal biological adaptive process. Now, what happens during hormesis? In hormesis, there is a dose response curve. Means if we give a low dose of any stimulant or a stressor, it, organism response will be very high, and it will be in a good side. Okay, it will give us a better result. But if you give it a higher side doses, means if you give the high concentration of the drug, it will give you a very pro inhibitory or inhibitory kind of a response. Okay, they will not allow a universal biological adaptive process to form. Means if you give a low dose, it will give a universal biological adaptive process to start. So this is what is happening. So hormesis is nothing but it's a stress response system. It's a drug of homeopaths which is involving in a stress response system. So functioning of the nanoparticle in homeopathy. So how the nanoparticles in homeopathy is actually functioning. Now this is important. If I'm already, as I already told you that nanoparticles are very toxic if you are using it at high concentration. And this is the smart way where drugs are used in a homeopathy. We are using nanoparticles actually, which are very much bioavailable, which are very active and reactive, and requires a very low dose of material, and that's why it is not producing any pharmacological or toxic effects as compared to the bulk form which we are using in a allopathic and other process. So that harmonic dose response is not going beyond no observed adverse effect level. Means we are not giving nanoparticles in the homeopathic remedies beyond no observed adverse effect level. So we are always maintaining a very low dose of nanoparticle, and that is with the no observed adverse effect level. Now, this is very important. So this point translates into the scientific plausibility of exceedingly small amount of homeopathic remedy nanoparticles being able to exert the biological effect in a cell or organism. So small quantity gives the better effect. So last point is this, whether homeopathic nanoparticles, which are typically delivered in a pulse or intermittent dosing, would act not by direct pharmacological effect on receptor, but rather by inducing self-propagating adaptive response. Hence, we are not giving nanoparticles or homeopathic remedies continuously. We are giving it in a pulse format. We are giving in a doses. As we know, we take in the morning, we take in the afternoon, we take in the evening, we take in the night. Most of the time, we take only 2 times. So this is an intermittent dosing of the regime, which will help to develop a pharmacological effect on the receptors, and thereby a self-propagating adaptive response is generated. And that helps to tackle this exogenous stressor or a danger signal in a better way. So this is the last point on this, is that what is the homeopathy is then? Homeopathy is nothing but the adaptive network nanomedicine. It is nothing but the adaptive network medicine. Now, how this is? Because it has been said that if you use the drug for the long term, it will give us the better result uh, in a short doses. While if you are using any high bulk form material in a long term, it will give you a negative effect. And that's why we should not take a drug on a high concentration and for a longer period of time. So this is what is the important point revealed by the Hahnemann, that uh, homeopathic remedies are actually working in an adaptive response manner. So that adaptive response is actually helping you to keep the immunity strong, to keep your hormesis balanced, and by which actually you are able to cure any disease, any ailment, any infection in your body, which is not possible whenever you are taking the bulk form. So in a summary, what we can say, nanoparticle cross adaptation sensitization model. This is very important. Nanoparticle cross adaptation sensitization model of homeopathy remedies provides an evidence-based foundation of comprehensive research program and new clinical approaches to the integrative medical treatment. We should focus, every researcher should focus on a nanoparticle side of the homeopathic remedies. Understanding the mechanism will help the practitioner to better understand when and how to utilize the homeopathic remedies for the patient. And we can now treat number of infectious diseases with this because of the presence of silica nanoparticles also. This is very important because they are now acting like a super adjuvant. So role of silica nanoparticle we should keep in mind while whenever we are going for the rigorous study of nanoparticles with the present in the homeopathy. So homeopathy is not a conventional pharmaceutical practice. Actually, remedies are not conventional drugs. Rather, as nanoparticles, homeopathic remedies are low intensity environmental stressors that the organism detects and threats to its survival. So whenever we are giving a homeopathic remedy, actually our body considers it as environmental stress and they respond. So the nanoparticles in the remedy augmented by silica nanostructures, and now they are acting as an adjuvant and also the non-immunological stress. So taken in together, what is our study? What is our findings tells us that a modern, scientifically more relevant, but neutral integrative term of homeopathy should be adaptive network nanomedicine. Means our drug, our homeopathic remedies are nothing but the adaptive network nanomedicine. I urge all the researchers to keep your focus onto the nanomedicine part. Drugs of homeopaths are actually the nanomedicines. Every research organization should set up a scanning electron microscopy, transmission electron microscopy kind of system in them. Whenever you are forming any new drug formulation, kindly check it out. What are the features of the nanoparticle present in that? And possibility is that in the future, we can give a very concrete dose formulation based on the nano drugs to cure the patient. Thank you.
Stop your screen sharing. Stop your screen sharing. Thank you. Thank you. Dr. Gore, please wait, because you have some question.
Yeah.
At the end of the lecture, our next speaker, Dr. Naik, will come with the question answer session.
Yeah.
You may go through the questions in the chat box. You have only one question. You please go through the chat box.
Yeah.
Next speaker, Dr. Naik is ready. I request Dr. Jyoti Gajbe. Dr. Nayak is from Central Council of Research in Homeopathy. Jyoti Gajbe, open your audio and video. Dr. Nayak is a senior scientist of Central Council of Research in Homeopathy. He is the Department of Epidemiology. I request Dr. Jyoti Gajbe. Can you hear me? Jyoti Gajbe?
Sir, I hear you, but I'm unable to start my video because the host has disabled it. The message comes, the host has disabled the video.
Dr. Karthik, can you hear me? Dr. Karthik, please allow him. Allow her, please. Dr. Karthik, can you hear me?
Yes, doctor, I've done it.
Okay. Please, Jyoti, open.
Yes, sir. Yes, sir.
Okay. Now, we please give introduction of our next speaker, Dr. Devdatta Nayak, from Central Council of Research in Homeopathy, New Delhi headquarters, senior scientist. I request Dr. Jyoti Gajbe from the Department of Community Medicine to introduce him in this platform.
So our next speaker is Dr. Devadatta Nayak, sir. He is MD Homeopathy in Matra Medica. He has done his MD Homeopathy in Matra Medica from the JSPS Government Homeopathic Medical College, Hyderabad. Now he is pursuing PhD in Public Health from the MIT University, Noida. Regarding his professional experience, sir presently working as a research officer homeopathy or scientist too at Central Council of Research in Homeopathy near New Delhi at Apex body for undertaking organized research in homeopathy under Ministry of Ayush and Government of India. He has also experienced in Biju Patnaik Government Homeopathic Medical College, Odisha. He worked there as a research associate. Then also worked as a Central Council for Research in Homeopathy as a senior research fellow. Also worked as a research officer scientist in CCRH only, and now as a research officer scientist too in CCRH. He is the recipient of Young Scientist Award of Central Council of Research in Homeopathy in 2017. He is coordinating fundamental and collaborative research studies. He is actively involved in studies on epidemic disease like dengue, Japanese encephalitis, malaria, HIV, and COVID-19. He has co-authored 38 papers published to his credit in peer-reviewed journals and 20 articles in no-reviewed journals. Sir also has co-authored a book. A book's names are An Update of and Bowel Nosers, contributed to many publications of CCRH, that is Homeopathic Science of Gentle Healing, Keynotes of Homeopathic Materia Medica, Volume 1 and Volume 2. He has also presented various papers in conferences, workshops, and CMEs. He is handling so many clinical trials as a PI and co-PI. He is handling so many clinical trials as a PI and co-PI. Some of them are pharmacological studies of homeopathic drugs, role of homeopathic medicines in cancer, regression and rejuvenation of depressed immune system, homeopathy therapy on duration of labor, for exploratory experiments on ultrasonic velocity in homeopathic potencies, action of belladonna, calcarea carp, and tuberculinum in Japanese encephalitis, and comparative cohort study to evaluate effectiveness on influenzinum in prevention of influenza-like illness. And his recent studies are the comparative cohort study to evaluate effectiveness of influenzinum in prevention of influenza-like illnesses. So, sir's today's topic is homeopathy in epidemics, which is evidence-based. So today, sir will going to share their experience with us. Thank
Homeopathy
you.
in epidemics or epidemiology?
Homeopathy in epidemics, evidence-based.
Evidence-based epidemic. Okay.
Yeah.
Thank you. Thank you. Dr. Nayak, please start your program. Dr. Nayak. Okay.
Am I audible?
You are fine.
Yes. Good afternoon, everybody. This is a difficult time. Usually, this made us learn many things in this COVID, and so anyway, I am sharing my screen so that I can share my PPT. My screen is visible to others.
Visible, sir.
Yes. Okay. Yes. This is regarding because you know, our previous learned speaker has pointed out some things, because we are working many things, and the major problem with homeopathy, when I joined in research here, I was also somewhat thinking what is happening, how it is happening. But the major question is that, can we measure everything? Do we have the knowledge to measure everything? Do science has the answer to all the questions? Can allopathic medicine, modern medicine, can they measure? Because I have interacted with around 30 collaborators. I think IIT, All India Institute of Medicine. We have our Besu from Calcutta. All the oldest and Center of Excellence institutes are collaborating with us. Many immunologists. I have asked many questions. How, when you are telling your medicine is acting, can you tell me your medicine is acting? They don't have that question, but it is a propaganda. Because when I am marketing something, I will tell this is acting. You may see in allopathic medicine, some drug comes, within 2-3 years, it goes away from the shelf. What happened? There was so many side effects. Whether they didn't aware about the side effect, they were aware about the side effect. And but the problem is that they have spent a lot of money, so they want to take many out of the market. So there are many things in there. because sometimes because when we are I do something, I have to sell something, I will tell the better part of it. So that was the problem of science. It is not of individual, it is problem of science. When science become, earlier it was science was the part of a legacy, all these people are doing, they don't have any expectation out of that. But now science is a business. If I am doing, I have invested some time, I have invested money, so I expect something come out of it. That is not happening with homeopathy, because the drug which were discovered in the time of Hahnemann's time, still it is continuing. And the easy way of making it, an individual who is somewhat intellect, he can make a medicine out of this. So that becomes, it is now no more supportable to the commercial part of healthcare. And that is the problem we are facing the most challenge. It is not science. If somebody really have true unbiased nature, he will always see homeopathy as effect. And the second part comes explanation of its effect. How many times nanoparticles are not new. Nanoparticles was there since eternity. Now we are able to see. So we have started asking, no, see, this is nanoparticle. Tomorrow our technology advance, so we will explain in a different way. Let us take into the example of molecular biology. Before 1980s or before Watson Crick 1960, 70, when they have seen DNA, we are also using medicine. We are also getting result. We are getting cured. But the explanation changed. The way of approach changed. But effects are always there. Human always uses effects. When we find let us take this situation of COVID-19. There is a crisis. We don't have medicine. Can you bring out a medicine within 2 days, 2 months, 5 months? So the term started repurposing. So science always moves with observation. It is not analytical part, comes the subsequent part. We explain the phenomena subsequently. Till date, we don't know how sunlight is affecting and how many ways it is affecting. We don't know. We know some part. Like a very good idea of that picture is scientist looking at the elephant. Somebody who has its trunk, he tells something. He has his leg, he tells something. So no, you will not find an answer to what is the elephant from scientist. You will find what I am doing, I will tell that. Let us proceeding to that. Basically, most of, I think, because homeopathy fraternity, they know CCRH, and this is apex body under Ministry of Health for the research and coordination. Now, gradually more things are coming to our part. This major part is to undertake research and to also promote research through collaboration, through other hand-holding. We also promote young researchers in level of undergraduate, postgraduate for a short-term project of 6 months, and postgraduate their dissertation work also support, but it is a merit basis. This is a network. We have almost presence everywhere. There are red part we don't know, we don't have, but we have collaborators. Like Madhya Pradesh, we have collaborators. Like Gujarat, we have collaborators. In Karnataka, we have collaborators, but we don't have any collaboration in J&K. We have many aspects. We are doing almost all aspects of research for homeopathy, starting from its medicinal plant cultivation, then standardization, proving. Those who are aware of this drug proving homeopathic part, they know that how medicine development is there. We are working on all aspects, including the fundamental research. And we have active collaboration with that Bombay, those who have started nano particle, but it was there was a story behind it. The first project was started in IIT Delhi, but the PI, what he was rigid that first I will get the second project, then only I will publish. That took us for around 1.5 years. In the meantime, IIT Mumbai people, they published it. So that's what a different story though, but it was started from we have started early, around 10 years before that. It was started. Now many part we are establishing a fundamental research laboratory at Kolkata along with the IIEST. That is its major activity will be only fundamental research to homeopathic aspect. So along with that, we are also having other diseases. So I'll just expect, because the topic will come to that topic. Infectious disease is a very dramatic up and rise. Sometimes around 1960, that at that time the Surgeon General of USA, he told, because there was advent of antibiotics are highly efficacious. So he told you should close the books for the infectious diseases, because there is no need to read it because we have just declared, we have just won the war against these microbes, but within 10 to 15 years, it was again the microbes have started raising their heads. That we have found out that there are drug resistance, then new microbes has been come out. So in the meantime, you have seen that there are a lot of microbes have come out like Ebola. It was there, but it is a resurgence. There are something emergence and resurgence. There are microbes which was there, like dengue. It was there before 1950 also there, but they are a very minimal type thing. But they become resurgent and their incidence or prevalence become high. Tuberculosis was very high, then it got gradual down. Now it is increasing again because due to drug resistance. And similarly, but the problem is of we are always being blamed. India has been always blamed for drug resistance. You may be knowing the super microbes that streptococcus, that it is highly drug resistance, and so all these things are there, and we are also facing the largest challenge in India in terms of tuberculosis, in terms of dengue, in terms of many things. We are facing a lot of change because we have a highly dense population in comparison to some of the Western countries, and the health behavior is different for us, and we are facing that to the add on to the our environmental problem, that is tropical country who have extreme heat, then cold, then humidity. So these things are very much uh, helping to propagate these diseases. But the problem is that we are being always blamed that we are bringing up the drug resistance of these things. That I have already told that we are bringing out many things, like we are hotspot for emerging infectious diseases, that almost there are variety of zoonotic diseases also coming in India. Recently, you may find out in Mumbai, they are coming up with another zoonotic diseases, vector-borne diseases, where a lot of problems are there. You know, if this disease also become a political issue for us, like dengue in Delhi, once it comes, then there are a lot of problems. This season has started, but because of COVID, this time there is less media coverage. Coming to this, what happened to this type of infectious disease? You see, this is since 1918, that Spanish flu. Around 50 million deaths were there. So there are, you can see comparative with there, and HIV, it killed around 30 million deaths in around 1981 and afterwards. But ultimately now this COVID has progressing fast. We don't know how far it will go. Though we are telling the death is less, the problem is of two kind. First, we are telling direct death, those who are hospitalized and dead dying. But second problem is that those who are affected and they are declared as they are out of disease and now discharged, they are also facing a lot of problem in terms of inflammatory diseases like cardiac arrest. Sudden cardiac death has been increased in Italy. The area which was most affected in Italy, they have seen a 70% rise in cardiac death in comparison to last year. So that means this COVID, post-COVID, is also taking a toll on our body, and that death, if it will include it to the COVID death related death, then the figure will be very high. Then comes this, how many deaths are there? So most death was in Spanish flu, that is 50 million people have died. So average age has decreased at that time. So there are many aspects to this death reporting also, because you know in our India also we don't have a good surveillance system, and so that's why we are unable to give an accurate figure, and different provincial government has different rules, different type of reporting, so that becomes a very difficult question to tell India how many are affected, though we come out with some of the figures. And these infections are very heavy burden. You know, this COVID alone only contracted our GDP by 23%. So that is huge economic burden comes along with this type of infectious diseases. The strength of homeopathy comes here, that when you come out with a new disease, then you are very helpless. Like in this, they have started with a hydroxychloroquine, then they have gone to the HIV drugs, then they gone to the anti-flu drugs, then ultimately they're roaming around here and there. But when it comes to homeopathy, there are always a criticism. How homeopathy? Where is the research evidence? But I question, where is the research evidence for hydroxychloroquine? Where is the evidence for your HIV drugs? Where is the evidence for your anti-flu drugs? Where is the evidence? There is no evidence. But because they are the mainstream medicine, they can do anything. But when somebody comes to take the share of market, then they started questioning. So this government has rightly started the activity that it is not that you cannot question other system. When you don't have a knowledge of other system, you cannot question the other system. The way of development of drug is different in your modern medicine. So the drug development is different in homeopathic medicine. When you don't know, how can you, and the testing, you cannot measure water with a solid with a liquid methods. So measurement of each is different. Orange is orange, apple is apple. So that's what it is. The problem is of the science is here is that it is become business. Scientists, most often they have become very narrow-minded. Narrow-minded in terms they look through only their own perspective. If I am of molecular, I look on that. The wider interest and the interest of public is very minimum, very minimum. The very most of our, even including me, when I am doing on this area means I will pertain to that. I will always look for this. But I will not look, see there may be better Ayurvedic drug may be there. But I am only interested, no. I will do it in business. It is become, I started doing a market. So I don't take the failure of my part as a failure. So that is the problem. But homeopathy has consistently shown a very good effect in different epidemics, pandemics, starting from Spanish flu. The first evidence, historical evidence or recorded data is started from Spanish flu. And that time, those who are, we will have a narrow, rapid go through this data. These are very observation data, but you know, the time period when these things are done, it was not a time of randomized control trial. It was done according to that time of that developmental stage of science. That was the post-Renaissance period when observational science was more and that experimental was less. So there are strengths. I should not go this. Before that, I want to tell really how much evidence-based is our modern medicine. This is the graph. You may see this is from BMJ, British Medical Journal. They published a clinical evidence book earlier. This graph was there up to 15th edition. But after 15th edition, when alternative medicine, our homeopathy, Ayurveda, they started quoting this graph. They removed this graph. My question is that, you see, only beneficial is only 15%. This 15% drugs exist in modern medicine has some evidence it is beneficial. Remaining 85%, 45% unknown effect. Out of that, 5% unlikely to be benefit. Then around 4% there is harmful effects. If you will take all together, it is 85%. 85% medicine do not have effect, but they are used. For example, simple example I am telling. We give antibiotic also because we also work in integrative medicine. We take project on that. In dengue, I have seen, you will see that. Also in COVID also, we give azithromycin, we give antibiotic, how it is effective. And you may have seen that recent data, hydroxychloroquine increases the chances of death in hospitalized cases, severe cases. So where is the evidence for that? But still we propagate that and we become very silent. Something comes out, we don't propagate that. We just become silent in that. But we keep on criticizing other systems. So that is the problem. So it is the time our doctors should come out with these things to counter them, to question the modern medicine. Where is your evidence? Where is your evidence? You just tell us where is your evidence. Just give new names. Personalized medicine, this medicine, that medicine, this part, the particle was there, this was there. You can make more nanoparticle in that. But the question is that how effective they are. How much effect is that? Nobody comes out with that type of data, but they comes out very short-term data of 5 months, 6 months, trial is being conducted 10 months. After that, we don't know. Even if you will take into consideration diabetic patients, there is no, at the end, after 30 years of a diabetic, there is no effect of anti-diabetic medicine with related to its death or morbidity and mortality. In they tell take calcium. Calcium has no effect in terms of decreasing mortality or decreasing the fracture rate in a long-term effect. But in a short term, we just see for 6 months, this is fine. So there is an effect. But when it comes to long term, we don't know how toxic it is. We don't know. Actually, I need not to ask take this slide, but after the discussion of our previous speaker, I took this slide particularly. I want to put in our young minds, our people, that there is no evidence. Medicine is always a trial and error. Since its eternity, trial error becomes that. And if you go to each individual doctor, they use their experience more than scientific evidence. They believe in their experience. You go, every allopathy doctor, they will change. You will see same effect in COVID. You go to state, Delhi state, go their for COVID, their panel, they have different approach. We have seen already because we have done trial in Maharashtra, we have done trial in Delhi, we have done trial in Madhya Pradesh. They have different standard of care. They advise differently. Hydroxychloroquine is not given in Delhi to the patients, but it is given in Madhya Pradesh. If you will go to Maharashtra, they have given it as a prophylactic. So they are different. Nobody believes in that. They go, the physician is always believes more. It is a Bayesian type of thinking that they believe in their experience. They take their experience into front. Then they see whether it will act or not act. And this is also true. Sometimes, for example, one of my friends fall ill here in typhoid. Another friend who is a modern medicine physician there in Odisha, we called him and he told give some medicine, but it was not going. Then when asked, we contacted here some another person. He told no, that medicine will not act here. Our experience says this, you take this medicine and he got cured with that. So that's what the personal experience, because those who are doing practice, you cannot replace a personal experience with a 6-month trial, 2 months trial, because you don't know when a person will go on taking a anti-hypertensive, anti-diabetic for a period of 10 years, 20 years, 30 years. What will happen? How other organ will behave? How other organ will be affected? No data. No, nobody can tell now. First question. Second question, when the trial is being conducted, the trial conducted for single disease and single medicine. I want to do a trial on asthma, that I will take patients of pure asthmatic condition. But when I prescribe that in the clinic, I prescribe a asthmatic patient, I give asthmatic drug, he may have skin problem, I give another skin drug, he may have diabetes, I give anti-diabetic. So what will happen? I mix a cocktail of so many drugs into that person. I don't know. Nobody can give that answer. But because the population of modern medicine, all the policy makers, like at the level of health policy makers, they are modern medicine. They just ignore that. You just question them. They don't have an answer to this. They just tell you this drug is effective this. But when I will ask a person 50, more than 50, they will have 2 to 3 complaints. When I will prescribe the 3 drugs which are never done a trial combinedly, how they are going to behave inside in my body? So there are many things. So I put this into our homeopathy brothers that don't go in there that this is evidence based. Evidence is not always a clinical trial. Evidence can have a personal experience, a case report, everything is evidence. That become a very narrow type of analysis, that this is not evidence. Evidence based is a very bigger area. It can have a observation study, it can be a single case record, it is a personal experience. Everything becomes a evidence based. Let us come to the now I put stop to that argument there, and because whenever you should somebody questions you, just put this question to them also. That's why I put there, because the time is here. We should look at the benefit of the public that is being hampered by some of the policymakers. They just want to question. Now in Maharashtra government, they have permitted ICE. Why so late? Why so late? The question is that when you don't have evidence for allopathy medicine, you allowed, but when asked for ICE, you didn't allow. What is the rationale behind it? So that we don't know. So that has to be questioned then. So Dorothy Sephard, I think almost our homeopathic part, our brothers knows brother and sisters, that it was written way back in around 1960 something. You see, there is no disease of present day disease. Like I will take dengue, it is not there. I will say chikungunya, it is not there. HIV, it is not there, because that was not so much prevalent at that time. So that means we need to relook into this, and we have restudy all these new diseases and put our experience to the use. So there is some historical part we will go through, and this is the first that in Spanish flu. No, recently there is a lot of comparison of Spanish flu, because it was happened 100 years before, so it is 100, and both the disease, they affect the respiratory tract, they do pneumonia, the ultimate death is due to pneumonia and all these things. Though corona had some more other effects. At that time, those who have received the homeopathic medicine, and they have only 0.7 case fatality rate, case mortality rate, and 1% around. But those who have not received it is 0.5 and up to 30%. It may be increased up to 30%. So there are many other diseases which have been taken. So you see typhus fever in 1813. There is also a less mortality rate. If you go to cholera epidemic 1831, it is again homeopathy has less mortality rate. But the question is, this comparison is not with the modern present day allopathic antibiotic. These are in an era when antibiotic was not there. And present day also similar thing is going to when antibiotic resistance is there. That means antibiotic is not there. Though it is there, but it is not effective. So it is also in cholera epidemic 1831 Austria, again 33%. And homeopathy in cholera epidemic 1854 London, still it is less. Everywhere you will find yellow fever, it is less. Everywhere you will find that homeopathy has a better result in terms of decreasing the death rate, all these things. Diphtheria, it is again, you see, 83% death, and homeopathy is only 16%. Meningitis prophylaxis, similarly, it has less in. Then coming to the recent origin, what happened? These recent diseases, like in terms of chikungunya, I'll start with the older one, and later I'll end with COVID, present day COVID, what we did. This was done, this trial we have done at in 2007, where a huge outbreak in Kerala, around 10% population was affected with chikungunya at that time. So we have given Brian Alba, as we know, we have a concept of genus epidemicus, where we select a medicine based on the presentation of symptom totality in the affected patients. So you can see around 20% risk reduction was there. It was a huge trial of around 42,000 population, and the overall the death rate was, incidence of that has decreased by 20% in homeopathy group. Again, coming to the leptospirosis, you know, this is, though it is treatable, but the ultimately when it becomes a huge epidemic, it becomes difficult to treat each cases. They come at a later stage. So this study was in Cuba. After this study, there was a renewed interest in homeopathic prophylactic, and because they have shown around, they decreased the incidence of this leptospira. You can see the after intervention, there is a forecasting of that. You can see how it was forecasted, but how it has gone. Historical median, it has gone beyond the historical median. You can see. Next slide I'm showing. You see cases of 2000, it has gone historical median. The historical median is that 2002 to 6. The incidence gone, though it was predicted before. It was predicted. It was predicted. You see so high, but after incidence, it has gone below down. Similarly, in non-intervention group, it has gone high. You see, though it was predicted, the way it was predicted, it has gone in that way. That means homeopathic intervention has decreased the cases of leptospirosis. Coming to the then dengue. You see, this trial we have done at a tertiary care hospital in Delhi. That is Hedgewar Hospital. That is a tertiary care hospital under Delhi government. Here we have given, as you know, in dengue there is no antivirals. I will just tell you some briefly about dengue. What is the evidence they have? They tell we are treating. They don't have antivirals. They cannot give painkiller, you know, because there is a bleeding tendency. Painkiller or this ibuprofen has a detrimental effect. But they just give, they replace platelet. That is not a medicine. They give antiemetics to prevent vomiting, and antipyretic. Recent evidence says if you give antipyretic, that is also detrimental effect because dengue also affect your liver, hepatic affections are there. So once you give paracetamol, again it damages your liver. So that means present evidence says if you give paracetamol, it will be detrimental rather than beneficial to the patient. So that means they don't have any option, though they treat it. But if you will tell them, so we have to give homeopathy, no, where is your evidence? The question is that where is your evidence? Everybody should start questioning them, because otherwise at the loss is public. It is not a loss of the allopathy or homeopathy or a doctor. The loss is to the public. The public is going to lose their, somebody will lose their near and dear. Somebody will become economically burdened because a poor person, once they are admitted, you may be seen this story of our from Gurgaon, that is hospital, the famous hospital. They have taken 15 lakhs. At the end, the boy died. But during this period, when you have done in 2015, there is huge outbreak in Delhi. No dengue patient died from this hospital. There are, when we have entered, there was every bed has three patients are lying. In every bed, there were charpais in between two beds, regular beds, means hospital bed. They put charpais. In each charpai, there are another two patients. After 15 to 20 days, when we have started, it was vacant. They used to discharge the patients around 60,000. Means when there is an increasing trend of platelet, they used to discharge the patient when they reach around 60, 70 and they have an increasing trend. But when you have started this after 15 to, they have stopped discharging because bed become vacant. So even persons have more than 2 lakh platelet count, they are not being discharged. So they just left the hospital. They tell it lama. So you just see the graph of platelet. H plus UC means that is homeopathy plus usual care. Usual care is that I already told you. If you have fever, you will get a paracetamol. If you don't fever, you will not get it. If you have vomiting, you will get antiemetics. Otherwise, if you will be stayed there, you will give a fluid replacement only. Fluid replacement is the only standard. In that case, those who have received homeopathy, you see, they have a platelet count increase in single day, 19,000. 0 means at baseline, and 1 means that is a minus. 1 minus 0, that is 19,000 increased. And you see it is only 7,000 increase. That means if you have only 1,000, you will have 20,000 by one day. That means you don't need also platelet transfusion. And this line, two lines you see, this is very highly significant. If you understand the statistical nature of this p-value, you see highly significant. And this trend continued up to 5 days because, as you know, once you survive from dengue of 5 to 6 days, then automatically your body will recover and you will become, your platelet count will become normal. This is published one. This is published in Homeopathy British Sampath Journal. Second part, they tell there is no preventive. They come out with a vaccine for dengue vexia. And if you have gone through the data of dengue vexia from New England Journal of Medicine, they have given in Philippines. The story is like that. When this was come, the physician who has done, the scientist who was a Philippine, when he did, they come out with this vaccine, then your Sanofi Pasteur has taken up this company, taken up this vaccine, they developed, and in the meantime, this fellow becomes secretary of health secretary in Philippines. They have started giving as a public health program to all the children of Philippines. After 2 years, the number of hemorrhagic dengue has increased in Philippines. Then by that time, the government has changed. Then they have set up an inquiry, and that fellow has been put behind the bar. Though he was the scientist, he then become the health secretary. So now he was behind the bar. Now, after that, that was the news in your Lancet. So think of these things when something comes, they market it. It was also in government of India was also considering to take a vaccine. They were behind it. They have taken up a huge surveillance of dengue antibody because that vaccine will be effective once somebody has earlier infected with dengue. So then it becomes silent. Once these things come out in public, then it becomes silent. My slides are visible to others? Hello?
Yes.
So that what we did here, we just taken up some of the JJ colony, in metro cities, the juggies are there, that urban slums are there. We have started this program around 2016 of to systematically establish, study the homeopathy in the prevention of this infectious diseases, particularly epidemic outbreaks. This we did it at Mayapuri slums, that is near our headquarters, and we have taken up some Equatorium percolatum to 12,000 population, and we have not given to around 3,000. You see the only fever how much decrease? The only fever reported. Forget about dengue, chikungunya, whatever it may be. The fever itself is only 5% in medicine group, when it is around 34% in control group. That is huge. Fever burden itself, the recent survey says people fall into the low poverty line. They go below to the level of poverty line. The major cause is dengue, at least in Delhi. Because once you are admitted in a hospital, they will keep you another 3 to 4 days. In government hospital, if you find it is okay, most often you will not find a bed there. And if you go to the some charitable, like still it is very costly. So you have to spend some of this. So 8% population, they they go below the line of poverty due to several illnesses. And the problem is that we in India, 1/3 of urban slums of world, they are in India. You see how much huge burden is this. Simply if I am able to decrease this disease, this fever, I can save the people not only from their mortality, morbidity, I can save them economically. I can save a major chunk of population. That is 1/3 urban slums of world urban slum. Total 1/3 of urban slums of world are in India. So that is huge population. So that's what, this is a very interesting data we have generated, but still the problem is that we are unable to still take it into the public health policy. Though somewhat in Kerala government they do it. We have written all these things letter to some of the state governments, all these things many a time, but it didn't happen. And if you see to the dengue, it is only 2% in medicine group, and it is 3.9% in. So because only 14 cases they are tested. Most often the people do not go for testing. They only do test. Those who have some severe, they go to the hospital, they test it. But subsequently we have taken up some of the trial where we want to do test, but still they don't give blood sample for testing. That time chikungunya, I think it was more. It is around 27% has chikungunya and 5% has only in medicine group. See the graph of that week wise. This is a graph which tells every week how many percentage of that population are affected with fever. You see every week in control group around 2 to 4% people are affected with fever. And when it is gone down, it is the period when it goes off around October end. It goes off automatically because there is a decrease in vector. This is a vector-borne disease. Once the vector mosquitoes are not breeding, so automatically the incidence of disease goes down. So what more evidence we need? These are controlled trials, all these things. But the question, once there is because these are population-based study. Once you do this population-based study, there will be question, where is the RCT? If you do RCT, you have this problem, that problem. But that is not question to. That same question has to put before your modern medicine. Your modern, almost everybody should be questioned in the similar way when it is questioned to the homeopathy or whatever eye system. We give this advisory, though we give it mostly in Delhi based, and but it is in social media. We put it on our website. All these things, we use all these things. Then comes influenza. Already I have told you that during Spanish flu there was data. It is effective. Also, we did this during the swine flu time. It is a multi-center trial all over India, wherever our trial there. ILA means influenza-like illness. Basically, what happens when a person is affected, most of the surveillance they are not, because it needs a BSL-3 lab facility to do a testing of this, and BSL-3 is not so easy at that time. Presently, of course, during some of the 4 or 5 years, a lot of BSL-3 has come up with that, but still it is very minimum to cater to the Indian population. So what they do, they just tell it there is a fever, there is cough, so it is an influenza-like illness. And the most often that 40 to 50% those who are with influenza-like illness, they are positive for influenza itself. So here we have given that as a homeopathy, and there we have seen that we have also started to do a comparison of our two potency like centesimal and millisimal potency. We have find out that both the potency are effective and very good, and against one tested a placebo control trial. So they are highly significant and significantly decrease the illness and recovery of the patients. Sometime we have also in 2009 we did this international survey, but ultimately other countries they could not contribute much. It was from India, it was contributed. So the most prescribed medicine was arsenic album. Remember why arsenic? This is one of the best for this time also. This time we have given arsenic. Here we have find out around arsenic was prescribed in 23% successfully treated cases are prescribed arsenic at that time. Next comes preventive study. Same, these are where this is not paper, that means these papers are under now process of publication. In the same urban slums, we have tried to test influenza genome. As you know, we have a medicine, homeopathy medicine. For others, homeopathic doctors, they are obviously know this. So you see the case rate. You see, if in medicinal group it is 17%, in control group how many? It is 32% overall. You see whether it is a coryza, cough, fever. In fever also it is 0.8 to 1.3%. If fever and cough it is 5%, it is 18%. This is ILI, that is fever plus cough. It is 5% in medicine group, it is 18%. So it is a huge impact data, these things are there. So the question is that there should not be question on this, because many a time they will require this trial, these many things comes out, but these are time tested. And you see this homeopathy has been questioned, but it is surviving. It is because of popularity. People want it. It is the second highest practiced medicine after modern medicine. It is practiced around 80 countries all over the globe. And in India, 100 million people use homeopathy itself. So though there are some people, they ignore that, but still why this propaganda is there from modern medicine? Because they know homeopathy is increasing. They are unable to counter it. In Kerala, you will see when we did a trial, they started writing against us. If you don't worry about homeopathy medicine, why you start writing? Because you never write about people, they are doing puja, they are going to temple. Nobody questioned them. Why you are going for a temple? Because you are suffering from a disease. There is no evidence. Once you are going to the temple or praying, you will be getting well. They are not questioned because they know with this nothing is good. But they know homeopathy is doing something. When somebody quarrels with you, why it quarrels? When they think there is a threat from this in terms of their market, in terms of their business, only then this thing starts. You see why there is a regular blogging against homeopathy in internet. Whenever you have opposition, always we should remember there is something in us. That's why there is opposition. If you are nothing, it is people who will reject you. There are many things come out during the human history. Many things come out, but many things has been rejected during the course of time. Then gradually the knowledge has evolved. But why homeopathy is not going out? Though there is no strong government patronage in European countries, because European countries they are doing legislative, because you know this modern medicine is a product of Europeans, and the pharmaceuticals are 15 major pharmaceutical, either Europe or Americans. The business of American is $9 billion in a year. So if when we will start marketing our drugs, they want India as a market. Evidence is nothing. They will always change the check post. Today they will tell you show this data. Tomorrow if you are coming doing research, you are doing fast and doing research, and you have reached there, they will no, we don't want this data. You just give us a genomic data. You are reaching there, no, you just give us that data. So they will change the check post. So that is another problem of our science, because we always look at the Western country. You see this is a check post for you proving you are doing a science. We forget as an Indian that we have also 1,000 years of science with us. We forget that. We never look at it. But China has preserved it. You see the effect of integrative medicine of China in COVID. The success rate was increased by 30%. the recovery was increased by 30% by giving their TCM. But that we are not learning from that. That are some of the experiences coming up with our Gujarat. They have given 50% of their population with arsenic and they have decreased the trend. Similarly, we also did in 1819, and you see the graph. You see, similarly, just see the graph. It tells many things. This brown color is of placebo group, and the control group, and this is the it is coming the incidence is this is the percentage of population, public or people, those who have fever in that week. It is not a cumulative data. Every week, this is a comparison to below the line. You see, it is only 0.01, 0.06, 0.04. It is 2%, 3%. It is huge things. So that still it is problematic. They take up at the government level when it goes. Many questions comes, but they don't question themselves. Why hydroxychloroquine? Why HIV drug? Why this azithromycin? Why XYZ drug? No questions, but question is for others. Similarly, we did another trial. This is a, this Japanese encephalitis is a highly mortality is very high, around 30% mortality. COVID just we are telling 1 or 2%, it may more 2-3% or 4% maximum. But in this trial, it was done at that you may be knowing this hospital at BRT Medical College at Gorakhpur. That is a medical college. They have Gorakhpur is famous for this. We had did this trial is around 2013 to 15. You can just see the result. The common formula for these diseases, if 100 child are infected, 30% will die straight. 30% will recover with a sequelae. 30% will recover without sequelae. So 30, 30, 30. This is a common formula. And if you see, death in placebo group is 29.8. That means it is following that same formula. What I told 30% it is from the textbooks. You see the same thing. In placebo group, what is happening? 30%. Here the placebo group is nothing is given. Everywhere in both groups, standard cares are given. I question then the standard treatment. What death rate decrease? If the standard care, they tell we have a medicine, we are able to treat them. Why this is 29 still persisting? The death rate should have been decreased to the 20%. At least with their treatment, it should go to the 20%. But when homeopathy was added, the death rate is 14%. 15% decrease in death rate. So these things are very encouraging, but astonishing that I questioned sometimes in some of the workshop to our director of health services of UP. I questioned them. What is the ways that to include a medicine in your SOP for treatment? He was not to answer that. When I repeatedly started questioning, I insisted on that. Then told it has to that central government has to tell that whether to include this or not, because there is evidence. This is published. This is published in a peer-reviewed journal. So the question is that we have evidence. We have a strong evidence because this is the one of the strongest evidence. This is the highest death rate. 15% decrease, means 50% decrease in death rate is big, and you see 30% more percent has a good recovery. That's what the question is not at this evidence. Evidence is there, but there is a systematic blindness towards homeopathy. Systematic blindness, ignorance towards homeopathy. This is systematic ignorance towards homeopathic system of medicine. Then comes how it is prevented. You might be knowing that in first earlier to the Gorakhpur, Andhra Pradesh was the worst affected with Japanese encephalitis, and my college there that become the center to distribute this medicine, that BCT. They have a mini pharmacy. They used to that manufacture do a polypacking of Baladno one color, Calcirac of another color, tuberculin another color. So that government has taken up this program for systematically for many years, and now they don't have any incidents. Some minimum incidence is there, but the same thing has been not adopted in UP. So that is the tragedy of our federal system, that some evidence coming, and this is the publication by that government of Andhra Pradesh. The data is from government of Andhra Pradesh, but still it is not taken up there. So how you will bring out what evidence you can bring? A government does there, publish a report, but the same another government do not adopt. The question is not with homeopathy. It is question with the policymakers and the other users, the other systems. They have to open up at the benefit of public, not at the benefit of that I am thinking, no, this I may have some different motives in that. We see the incidence rate. It was in 91. How it was there. incidence and mortality. And when it was started around 99, you see 2000, 2001, it is gone down. It is gone down. It become nil. Now also there are some people, they know, they do it, and this was done through not only homeopathic department, it is done through all the primary care doctors, whether allopathy, homeopathy, ayurveda, they have to give this drug to the public, that the children. Similarly, earlier to that, we have generated this evidence at Gorakhpur also, but nobody taken this result, never used by the public, by the government. This was done in Gorakhpur itself in 1991. Coming to this COVID, now we will end with this COVID, some of the slides. COVID, as you know, already pandemic has been declared around January, and you know already 10 lakh deaths are there all over globe. So 1 million deaths. So maybe they are going more. I don't know how it is going to happen, but this is actually deaths of that hospitalized death, and how this indirect death, that is death due to other complications due to post-COVID infections, how it is going, we don't know, and whether they will, how it will add up to this. And now Asia become epicenter. Asia and this American, now China has gone from that phase. We don't know. China is a very mysterious country. You see this middle one. Earlier it was very negligible. Now South Asia also contributing significantly. India particularly contributing, driving the incidence in Southeast Asia. And you know, it crossed 64 lakhs, and death also 1 lakh has crossed. I will compare this Spanish flu already I told you, but what we should learn from this? Because at that time nobody has done a trial, neither homeopathy nor allopathy, but they started criticizing us. When they started criticizing of that time with a modern knowledge, you should criticize that era with that era knowledge. You cannot compare 1900 with 2000 knowledge, because at that time the experimental designs are not very good, statistical tools are not implemented in medicine good in a better way. But still they started criticizing of in a publication of 2004. Why they are criticizing then now? And historical data, how it can be criticizing now? When we're not sure about how that has been done at that time. It is the data of them only. It is from a book that it is a very historian, medical historian. They only told it is 28%, 2% of that regular physician have studied. So you not should have that. But that is a way of that is a hospital death. It is a hospital death. Hospital death means those who are hospitalized. Like in case of our COVID, if we will take all the cases of 64 lakhs and we will compare the death, it is very negligible. Or if we will take the all seroprevalence data, then it is very 0.00 some death is there. But the comparison is with hospitalized and death. But now they questioned this in the recent publications, the recent past publications. So what happened from this? We have also drawn some of our lessons. When this COVID started, it is Ministry of Ayush. Because of Ministry of Ayush, we got an opportunity this time to drive our system. As you know now, it is one of the major, Ayush is a major discussion point of many government, at many agency, many NGOs come out with many things happened this time with the which never happened in our history. Not also during Spanish flu and the swine flu. So as soon as it was came around 30th January was the first case, 29th with the base of analysis of the data of patients of Wuhan, we have given arsenic album as a medicine, and that was given by Ministry of Ayush. But the game changer is this gazette notification, where they allowed us undertake research and with consultation, after consultation, because you know any clinical trial, DCGI, CDSCO, they come into play, but they only allowed, no, Ayush can be allowed because they don't have a new drug, they have already pharmacopoeial drugs, so they are already using this pharmacopoeial drug in day-to-day practice, so they can be allowed, and they allowed, and that only changed the scenario. And you will be astonished to know that 65% of clinical trials of COVID are from Ayush. That never happened in history, never happened during swine flu, never, because they never allow us to take up trials. It is not that Ayush cannot do a trial, Ayush personals cannot take a trial. They do not have resource. Of course, we have a limited resource, but this time, because of this provisions, we have already taken up around 65% clinical trials are from Ayush alone. Remember, and you will go to the clinical trials of other side. They are not clinical trials. There are many trials on disease, clinical epidemiology part. Medicine trials are very few in that in allopathic medicine. Next comes another game changer that Ministry of Ayush published guideline for homeopathy practitioners for COVID, where they allowed the personals, homeopathic doctors to practice. It is not for homeopathy; it was given for all the Ayush systems. So that our doctors become strong in that, and now based on this, gradually the many state government has allowing to treat, like Delhi allowed, in Madhya Pradesh they have allowed. We have done a trial in Madhya Pradesh. In Pune, we have done with some of the collaboration with the trial there. In Gujarat, they have allowed to treat all the cases with homeopathic medicine, homeopathic Ayush medicine. Coming to this disease picture, it has a wide spectrum of COVID. So because I am telling you, because I will relate our studies already what happened, we have already taken to this phase of the disease. First phase is 80% is asymptomatic. Out of 20%, major portion is mild. Then 5% are moderate. Out of 5%, within that and 1 to 2%, they become severe. And this is what we have done at that time to advise that common those who are having, they should take advice by Ministry of Ayush. They also take arsenic album. Those who are not exposed also as a prophylactic. Those who are exposed but asymptomatic or quarantine, they are also advised to take arsenic album. But those who are mild and moderate to severe, they can take a add-on homeopathy. So accordingly, we have before that, I should tell that what happened with that research. COVID is very minimal. There is no research publication. These are not a research experience publication. These are some of the experience one published. Second thing, from Mumbai, one of them shown that Mercurius albulis can be a genus epidemicus, but their systematic research has not been taken up with other than India. In India alone, we have around 19 research trials has been taken up. Out of that, you see, we always question that how all aspect has been taken up. That treatment trials also taken up, prophylactic trials are taken up, other type of data generate like survey, all these things also taken up. And major part is treatment. 53% trials are treatment trials. Around 19 studies has been taken up by homeopathy. Now recently some trial may have added up because every 1 month, 2 months, there were new trials are being adding up. We have also added one trial now from CCRH, and survey type of thing. Another trial is also in plan, 2-3 trials under plan. So most the studies are randomized control trial, because always they will question you why, if you do some graduation study, some post study, why it is not RCT. We don't believe it is not RCT. That becomes somewhat, we also become cautious than that. So you should try to take up randomized control trials. So that's why more trials are now randomized control trials. Then coming to the treatment trials, majority of treatment trials are randomized. Around 70% treatment trials are randomized control trials in COVID. And these are the drugs which have been taken up so far as yet for treatment trials. Like my major trials are with totality. And arsenic also taken up some of the trials, antimtard, urania, camphora, crotalacetus, eupatrium, gelsinum, zucamloritum. And 1 or 2 trials with combination. Then coming to the prophylactic trial, again the majority of prophylactic trials are taken up with a randomized control trial, and 37% of the study. And the drugs which are being tested for prophylaxis major is 75% trials are with arsenic album, as you know, because this is government has advised. And then comes Brineia alba, coronavirus nodule, somebody has taken, but that was legally not sustainable because they have not taken appropriate permissions, all these things, because this they have put a strong rider on taking of studies with corona COVID virus. So another camphora is around 2-3 trials, sarcolactic acid, and some of the combinations. So what we did, what CCRH did, because as you know, always there is expectation that is affects body, how it is performed, how it is moving during COVID, because all the advisories, we are behind that. We recommended arsenic album to the ministry, and it was given through ministry to the public. So behind that, it is the arsenic CCRH. Second, you have seen the guidelines for the treatment guidelines. It is CCRH we have drafted, then we have given to the ministry for finalization and bring out to the public, because ministry is legally binding, because ministry has the stakeholder, has the authorized authority to bring out such type of advisory, because CCRH is a scientific organization where it can give a evidence to government, then government has to take a decision. We did some of the trials with as a treatment. First we should see, we have done a prophylactic trial. It is a multi-center trial which use 32,000 sample size. Almost all metro cities are included in this: Delhi, Mumbai, Chennai, Kolkata, Hyderabad, Surat, Machilpatnam. Machilpatnam is a small part in Andhra Pradesh. And we have already enrolled almost 32,000. We are analyzing. And I can, because there is a rider from Minister of Vice now, because you know, Minister of Vice has constituted a task force committee. And in that task force, they have done, before anybody going to publish any trials result, they should submit to them. So we have given the interim data, and we are in the process of finalizing the paper. So once they will agree to go ahead with publication, so it will go ahead. Yeah, or they may be going, like you have seen in their Facebook, they are coming with some of the videos, all these things. How they are coming up? Because of this data, this study data, they are coming up with a social media activity. That is the data provided from these studies is enabling our ministry to take up social media campaigns. Another study we did, this was the first study we did on prophylactic when around April, we started first week of May in Delhi. Once we got the permission from Delhi government, we have started in the containment area. We have taken up around 10,000, and this data is encouraging. Both the study has similar type of result, and arsenic is a good effect on preventing over a period of 9 weeks. Of course, we are still following those who have taken arsenic, because you know, arsenic is never a vaccine, because always they question other side. I have taken arsenic 2 months before, and now I am COVID positive. I am to tell here, arsenic is never a vaccine. And what we got the data from, you need to have a repetition around 15 days. 15-20 days you need a repetition. So next comes asymptomatic case. So we know now we have given preventing, then there is a part, it's 80%. So we have taken one study at Bhopal Government Homeopathy Medical College. That become a COVID care center. So they are government permitted to give homeopathy medicine. So this trial has been taken up there, around 200 sample with all the 3 drugs and 1 placebo group. Then we have started, you know, this you may be seen sometime Rajan Sankaran, you are telling we will take trials. He also approached us that you provide, he got permission from that hospital. Then he approached us to provide technical support, provide protocol and how to conduct all these things, everything we have provided them. And this trial already they have crossed around 200 patients they have recruited. So this data interim we have not analyzed. Another with Agra, you may have seen Dr. Gupta. He has given some time that we are doing that. We have also provided to them this protocol or this, but they could not get sufficient number of samples. So the study could not progress now. So it has almost held at 49 patients since 2 months, 2-3 months. So mostly they will be unable to achieve the sample size. Another study we have done at Chirai Hospital. This we did very fast. We started in July. We finished the sample recruitment by August end. So we have done an interim analysis. It here also we got a good result in this. Persons those who are receiving homeopathy medicine has a better effect. But we cannot, data can be shared only after this government gives a permission that this paper can be go ahead with that. But we have submitted this data to shared with data to the Ministry of Ayush. Another study where we want to see that those who received homeopathy prophylactic, once if they fall ill, what will the outcome? Sometimes some medicine, though they fall ill, but the ultimate progressive disease are modified due to medicine they have taken. So this is a huge sample pan India. We have almost all 26 centers of CCRS. They are working on this. So almost all over around the India, we are collecting data for, we'll collect around 5,000 data. Those who have infected and recovered, if they have taken homeopathy, how was the outcome? Based on all these things, we have now come out with this, that if you are unexposed, but mostly now you have many people, 30% people are affected, so mostly they are exposed. So even if you think you are not exposed, so still you keep taking arsenic. If you are thinking you are exposed and quarantine, you want to quarantine, you take arsenic. In asymptomatic also, arsenic has given a good effect, so still you should take in asymptomatic stage. You are positive but asymptomatic, take arsenic. And uh, with mild COVID also, take as a add-on homeopathy can be given, and with moderate to severe COVID-19 symptoms, as a add-on homeopathy also be recommended. Coming to this at the end, one or two slides are there. I want to say that it is WHO. In 2014, when there is Ebola, uh, they are unable to bring out with any medicine, then unable to bring out some vaccine. They only said that it is ethical to offer an unproven intervention with a as yet unknown efficacy and adverse effect as a potential treatment or prevention. Homeopathy is not unknown. It has been given. It has been given to these drugs are pharmaceutical drugs. These drugs are published by Government of India as a pharmacopoeia. Still, we are facing some of the challenges, though there are some challenges have been overcome by Ministry of Ayush, but still there are many state governments has not incorporated directly to the COVID care and all these things, though we have demand is going up. And till that, we don't know how vaccine will be. As you know, this virus is a very tricky virus. They change very rapidly. There are many genetic changes or modification mutations have been occurred to these viruses. And another problem is that we don't know how vaccine effective is that, because already those who are infected and recovered, the half-life period be this antibody is only 36 days. By 36 days, 50% of their antibody is perishing. So that means if I go by that, take us 100 days. Within 100 days, I will not have any antibody. So that means I am again vulnerable to the infection. If I give again, taking the same philosophy to the vaccine, I get a vaccine. So how many days I will have this antibody after taking a vaccine? So 100 days, I need to, so I don't know. Maybe they will come with a better vaccine, we will have more protection. We don't know. These are things, some of the pictorial we have visited our doctors during that lockdown period, strict lockdown in May. They visited to the, you see, door to door, they have visited, provided medicine, then we have collected data, and over telephonic we are still following them till date. Over around 40,000 population we are following them in Delhi alone till date. This is in some of the Bikaspur area we have visited. That block was declared. You see, nobody is there outside except police personnel, those who are on duty, and all are inside house. That was a containment area where clusters of cases has come out. And this is early lockdown period when they used to block the house. Now they are not doing blocking house. They just keep sitting at somebody. People are there being asked not to go outside. They are blocking roads. But here they block the coming that building outlet. These are our doctors. They have visiting and they're giving that medicine. They used to give this type of yellow. So after all, I want to tell CCRH has, we have established epidemic cell in CCRH to have take this type of studies and to generate evidence for this. There are systematic research taken up. There are systematic, not only clinical, I have just told here clinical. We have taken up all preclinical studies. We are developing nojodes. We are taking up preclinical trials on effect of these nojodes on those cases. Also, some of the known drugs in preclinical studies. We have collaboration with NCCS Pune. We have collaboration with, in fact, we have collaboration with KGMU. We have collaboration with Punjab University. We have collaboration with NICED, National Institute of Cholera and Enteric Disease. We have our own virology laboratory. We have a BSL-2, Biosafety Level 2 virology laboratory at Kolkata. We have our microbiology laboratory at Noida. So we are coming up, we are planning according to the demand of need of the science, and either through intramurally or through. And other than that, these are things we have also taken up, planned the trials with influenza, malaria, Japanese encephalitis, diarrheal disease. So these things are going on. Already we have conducted around during last four years, 13 trials. And during for this COVID, there is a special issue come out with in our IJRX, Indian Journal for Research in Homeopathy. This is a free journal, online free journal. You can go through it to see how an epidemic homeopathy has done. That is basically on historical evidence of epidemics in homeopathy, not COVID, because at that time COVID, it was in March issue. So research had not taken up that time. So in future, we will come out with this type of publication of this study. Hope after getting permission from Minister of Ayush. And one of the article was there that you can see almost all infectious disease, whatever evidence studies, clinical trials conducted. So it was a review paper in that. And look into that our Facebook page. There are updates on this public social media regarding campaign, regarding medicine advisory by Minister of Ayush. Thank you all for your patient hearing. Any questions? Am I audible?
You are audible.
Yes, yes.
We have some question. And Dr. Karthik, can you hear me?
Yes, I can hear you.
Okay. So we have some question. We are going through the question for Dr. Gore and as well as for Dr. Nayak. I think Dr. Gore, have you gone through the question by Dr. Subhamoy Ghosh from Calcutta? Dr. Subhamoy Ghosh from Calcutta. His question, please go. Do you have any original work of your own with ACM and TM, FM related to nanoparticles of homeopathy for producing reproducibility of the work done by Dr. Joyce? It will be Dr. Joyce Bellery and Dr. Rajendran and Dr. Bellivarity. And so these are the questions. Are you hypothetical? Model done by different researchers. So what is your own contribution to homeopathy? This is a question by Asus. I think it is the question done by Dr. Exactly it is nickname or Asus. My suggestion to all the viewers, please join with your own name because we are going to supply the certificates. We are within the next day we are able to supply. Those who are joining by code name or by their phone, it's very difficult to issue the certificates. So I think I do not know Asus, who is Asus. Once I saw that it is Dr. Subhamoy Ghosh. Subhamoy Ghosh is question that is Dr. Naik's question. Dr. Gore, please reply your question.
Actually, we are working with scanning electron microscopy, transmission and others on the homeopathy, and not only that, we are also working with the plant nanotechnology. The question is that what is your real original work? Actually, our company is a contract research organization. So we don't publish the results, whatever we are getting, we are handing it over to the customers. So actually customers funds us for the research. So whatever the research we do, we hand over the data to them. And that's why we are not publishing the research articles. We have our own research articles, but not related with this because of the contract research organization that we are having. Another question asked is that of, I think, how to maintain the action of nanomedicine and it should not be nullified. Actually, we are working on a nano drug concentration, how to concentrate the nanoparticles. And that is a very important aspect because we should know what is the exact count of the nanoparticle per microgram or per ml of the drug or the whatever the composition what we are preparing. So for that, you should work on a concentration part. So we are using centrifugation method and precipitation method of the nanomaterial. You can check it out by some papers onto that. So that is what is the point where you can work on a nanomedicine.
Okay, thank you, Dr. Kohane Madam. I think you are satisfied. Kohane Madam from my college, Dr. Nilima Kohane, and Dr. Asus and Subhamoy Ghosh has put down some question to Dr. Devdatta Nayak. Subhamoy Ghosh, or Asus Subhamoy Ghosh. Dr. Devdatta Nayak is promising researcher of CCI. Nicely presented work today. My best wishes to him. Thank you, Subhamoy. And let us share recent important works of clinical fundamental of COVID-19 of CCRH in a nutshell for the audience. Dr. Devdatta Nayak, you please.
So there are many things are going on because, as you know, there are sometimes it becomes a IP issue. But I can tell we are very active in some of the areas like malaria, HIV. Also, recently we have taken up TB because we don't know, because this COVID has a very difficult time is coming up, because due to COVID, how COVID will going to happen, how what it will do to the HIV persons, how it is going to affect the tuberculosis, already compromised respiratory compromised persons like tuberculosis. So we have planned and we have taken up some of the studies on that. We don't know how far it will take up. And post-COVID, as you know, I have already not shared that, right? There are 40% people, those who are affected with COVID, that they have recovered from COVID, has at least one symptom. And around 30% they have two symptoms, persist for at least for 2 to 3 months. And children's, they are telling that they are not affected by COVID, but they are giving more inflammatory changes. And type 1 diabetes has increased in children. In UK, recent study says that around type 1 incidence of type 1 diabetes has increased double in comparison to last year. And how this is going to unfold, we don't know. And third thing, in fundamental research, we are working, so establishing some of the laboratories. Somebody has questioned at IIST. We have a lot of plan there, and starting from physicochemical till to how it is acting at a cellular level, how it is a very sophisticated lab. It is a highly sophisticated lab. That much I can say now. And we are also a lot of collaborations, like we are working on some malaria. I cannot tell some of the drug on malaria. And we are planning some of the prophylactic trial on malaria in Tripura, where we are looking in the tribal belt of Tripura. We have visited there. Everything was finalized. We visited that area that is around 120 kilometers from Agartala, at a very difficult terrain. And but due to COVID, it could not initiate. Of course, everything was finalized. Everything was on place. Recruitment was there, manpower, everything was. We visited. We interacted with their medical officer, and you will be happy to know that in Delhi itself, we have. It is not that arsenic we are doing trial. We are also doing on influenza trial. Actually, we used to do influenza trial every year from January to March. But when this COVID started, we extended the trial till now, December. I have not discussed on that. We will be coming out with more data on that, because you know public health trials take some time. We need to very confirm about this result and on the confounding factors. Third is that we are working on IIT Mumbai on potentization, how this succussion is affecting, how really if you give number of more succussion, so that it will be more effective. If you give less succussion, it will be less effective. And we are working some other areas of neuropathies, and some of the areas coming, it will come up, some of the publication under consideration on nojodes like malaria, leishmania, and some of the dengue nojode. We are working on toxic effect, because everybody says there is no toxicity of that. That also we are working on nojode, toxicity of nojodes, immunogenic properties of nojodes. So all these things are there. It will come up, it will become public, because you know these things are public funded. Ultimately, it will come to the public. But sometimes Government of India is very rigid now that being you are public funded, you have to come up with the IPs, intellectual property rights. If you are not coming up with patents, all this, earlier we don't have that type of provision that we should look for patents. Now we are looking for patents for long-term scientific credibility of CCRH, because they are asking if it is public funded, what knowledge you have created. So that going on, and this type of things are very ongoing process, and you maybe find some new things within another 5-6 months. So every time it goes on adding to that original things. Already there are around 10 or 12 collaborations are in active stage. Studies are with the active stage. There are planned around, because due to COVID we could not initiate. There are around 10 to 12 studies still under already cleared, financially cleared to take up the studies.
Hello.
Yes.
So
Any other question?
yes, please you see your WhatsApp. Please see your WhatsApp. Just I forward a message from Dana Wilmer from San Francisco, California. Dana Wilmer from California. Just I forwarded it to you. He has sent the message through messenger to me. California Dana Wilmer is a very popular in America. Just about you wanted to know about the publications that are done by the government. Do you happen to know if the government of India?
No. See what happens. The publications, it comes in terms of journal publication. They are published in journals, and these are published. You can see some of the publication is in our IGRH, and you can see homeopathy journal, that British homeopathy journal. Recently 2 papers are already accepted for publication in ahead of publication. One is on leishmania, another is in cancer. We did a collaboration with Amity University, and you will find some of the articles in cancer letters, oncology letters, some of the major journals it is there, and on Thuja, on sulfur, on Kalkirakar, and we are still in process. Some publications are under process. As you know, it will become very difficult to publish in a mainstream journal. Once you give a name of homeopathy, you submit by evening, morning they will reject it and send it back, because there is a systematic campaign against homeopathy. There is a systematic campaign against homeopathy, because see, there are things. If I am submitting an article, at least there should be some comments. You should reject based on a comment, but this is outright reject. Today give and tomorrow morning it is not there, and it is not there. There is a new author is submitting. They are outstanding author of standing for that 30-20 years. Same journal they published in from that laboratory, but when the homeopathy intervention given, they just reject it. So there are
Should I refer all the publications to him as a government publication?
actually I know he used to follow all these things CCRH. Once we have called him also. I met him at London also.
Program. I know that.
So he knows. I think there is no report comes in terms of government. One publication is there, that is from Minister of Ayush, that is homeopathy gentle healing. That is also research publication. This is that is a review type book type. So in that, what research taken up during last 10 years is given. That I think we have provided him one copy at that time.
Okay.
And, if he want, he can visit to our website. He can download that PDF. Okay. That PDF is free. That is freely available on our website.
Already I have informed that please contact with the DG in these regards.
He can write a mail to DG.
Already I have informed.
Email to Director General.
I have already shared the link of today's program to him. He is viewing this program from America. Personally, I share the link, and probably they have some World Homeopathy Summit from India. Rajan Sankaran is and Farooq J Master is going there. I probably that will be a virtual summit. So many people are coming. I think they will. He will publish. He will present this one informations in this summit. So thank you, Dr. Nayak. I really agree with you what you did.
One question is there from Dr. Parameswar Vishwal.
Dr. Parameswar Vishwal, what steps CSIR is taking to avoid the homeopathic practitioners about his research activities, Dr. Devbrata Nayak. Please reply on that point.
The question is big because CCRH not have that type of access to the practitioners.
Yes, yes.
First question is that the state government should come up with doing such things, so that we are always happy to be part of that. But the problem is we cannot organize it because we don't have that type of huge resource to manpower to put into that. Because if you continue doing this, so we don't have that type of resource. When the scientists will be busy with doing that, when they will be taking up the project. But we can always disseminate this if there is such type of like you are doing this type of webinar. You have asked the can CCS participate. Similarly, one PG then from Bihar they asked. So we are always to do this. That is our job only. We are paid for that to disseminate the research results, all these things. But such type of platform like this you have created, so it can be we are. They can write a letter to DG Director General.
I think
Automatically.
our college is the first college in India who invited most of the scientists of CCRH.
No, one from Bihar, I think some people they did.
One day they have organized. Now we are organizing up to 31st December. Most of the CCRH from the different part of the country are coming to give their lecture presentation, what sorts of research are going on throughout the, at least the people should know, our students should know, they will be encouraged from your informations. That this
Definitely, because many people are, I find they are discouraged when they are being questioned. Where is the evidence? What is happening? Why it is given? So sometimes there is a offensive also a defense.
No.
Sometimes so there should be offensive defense also there.
Told you in your lecture, I support you because 100 years back, Dr. Dana, that is the Doctor's Dilemma by Bernard Shaw. Please go through the book. The Doctor's Dilemma. What he told, the fashion of epidemics. The epidemics, the psychology of fashions becomes the pathology for the cases of every year of being genuine. After all, or induced epidemics. After all, induced epidemics, proving that epidemics can be induced by tradesmen and therefore the doctors. Even their statistical illusion in respect of their vaccination claim and perils of vaccination. So many topics he wrote 100 years back. 100 years back. Now this is happening. What you told earlier, that it is actually the businessmen, they are controlling the total health sectors. When there is a question of some sorts of how it works, but it works, it happens. So thank you, Dr. Devdatta, for your nice presentation, information. Dr. Deepa, Raje, Deepa, thanks and gratitude. And Dr. Dilip Gore, okay, now we are at the last part, sir, please wait. Your informations are very informative to our students and to viewers, those who have participated. Dr. Barbat sir, please open your mic. Dr. Barbat sir. Viewers, Kartik, can you hear me? Dr. Kartik?
Yes, doctor.
Open your video. Barbat sir, open your video. We have information. Tomorrow we are going to another webinar at 4 p.m. Please, my viewers, we have one scientist from CSIR, Niri, and he will speak on health hazards on air pollution. He's the chief scientist of Niri, toxicity and health management sector, head of the department. He will come at 4 p.m. Subsequent to his lecture, we have a popular speaker, international speaker, Dr. Ojit Kulkarni will be there on Mathemedica drug relationship. So I request department to Mathemedica, Dr. Pohane sir. I think Dr. Pohane sir has left. Dr. Pohane madam is with us. Dr. Pohane madam, you please inform Dr. Pohane sir to come on that day because of his department's topic. Head of the department, Dr. Pohane madam always remains with us. Maximum number of teacher has left, but still Pohane madam is still with us. So thank you, Pohane madam, for your question. I inform Dr. Pohane sir to come tomorrow at right time. Okay, thank you. And tomorrow's program and another information, please join with your proper name, because we are using some codes, using some your phone number. It is very difficult for us, very difficult for us to identify you. Please come your mail ID. It will be very, because we are planning already. We have planned to issue youth certificate, that a Karthik is preparing that one, and next time you will get the youth certificate through your mail ID. He will generating one forms, and we will upload in this all groups, and you please join with your phone number and join with your mail ID and for identity. Otherwise, it will not difficult. It will very difficult, and all the speakers will get certificate, no doubtly. So thanks, Dr. Deptanath. I want to say my compliment and supplement to the CCRH, because once it was the principle petito. It is the principle that what is to be proved, and that is also the first. It is stated. It is a general philosophical term. One arsenic, there was so much criticism came from different corners, even from the part of the homeopathy too. But what it proves, the two state government, what I told earlier in front of our director, really it proves that arsenic is doing well, our scientists are doing well. It proves that the CCRH is doing well. So many people, so many teachers, and criticize that how would be the approach, because of that it contrary to the organon approach. And therefore now it proves that really it works. It is the question of foresightedness. It's a question of credibility. What research organization, according to the research work, they came into conclusion that arsenic would be the medicine, and it proves that now it is not the principle petito principle. It is proved now that it is arsenic good. So think well done, and again I congratulate Dr. Devdana personally. I invite you to come here, and again in future we'll invite all of you, and this link will be forwarded to all the colleges of the India, that they should know that what we are doing with the CCRH. And also I forwarded different groups, especially the I Love Homeopathy page, comprising of 250,000 members, different part of the country. They are viewing these programs. That's what CCRH in India. Dana will say that only government in the world, only government in the world, because Dhanavendra came to one webinar with us, that only government through the world that doing well in Indian government today, through the COVID and treatment, and CCRH team is doing well. They praise it in my workshop. So really, you are doing the very praiseworthy work. Everybody, though believes the homeopathy, and one people China, from one people Julie Sung regularly join with us. Julie Sung is a close contact with the so many people of India. He joined with us from China, southern part of the China. He is listening. She is listening my lecture. I requested her to communicate your government to do with homeopathy. She told, no, it is very difficult for me. And once he has a plan to organize a seminar at Hong Kong, and because Farooq Master is related to them, and then they will invite the CCH people. Thank you, Dr. Devdatta Nayak. Thank you, Dr. Gore. And Dr. Another information, you please wait. Another information, all the viewers, in the next week, probably it is fixed now. It is on 9th October. You're coming with an exclusive interview with Dr. Rajesh Shah. Dr. Rajesh Shah, Bombay, exclusive interview with the people of the Antarbharati Homeopathy Medical College. Anybody can join with the Raj exclusive interview. It is the first time in India that Antarbharati Homeopathy College and Bahula Foundation Research going to exclusive interview with Rajesh Shah, very popular name at the field of homeopathy. Once he was praised by CCRH, and on 9th October there is an important meeting with CCRH with him. After completion of the CCRH meeting, he will join with us. He has committed to join. He has already forwarded his biography, and only the official invitation letter is required. I will request Dr. Barbot sir to issue the official letter to Rajesh sir. Rajesh sir is coming on 9th October. It will be a very important and exclusive interview with the Antarbharati team and with Dr. Rajesh sir. And obviously, it is the credit goes to Dr. Karthik and Bahawala team. They are giving scope and using this platform to inviting so many people of this country as well as from the abroad. We are inviting and spread the dissemination of the homeopathic teaching, homeopathic philosophy, and homeopathic learning in this point of time. Now the validatory session, Dr. Barbat sir, please open your. So many people from CCRH. There are so many people. It will take time putting them from Imphal, from Manipal, from Siliguri, so many people from Roja, Dr. Roja from Delhi, and so many people joined. Thank you to all of you. Please be with us. Please stay with us and with the near succeeding seminars because so many scientists are coming. Thank you. Please join tomorrow. It's 4 p.m. with Dr. Krishnamurthy and Dr. Ojit Kulkarni. It will be very enjoyable lecture of Dr. Ojit Kulkarni on tomorrow with the drug relationship. Every student should take part. Bharwat sir, please go through the validatory session. Bharwat sir.
Yes.
Yes.
Good evening, everybody. For both the speaker, Dr. Dilip Gore sir and Dr. Devnath Nayak, they are really giving us a good informative lecture. And every student, those who are as beginners in homeopathy, they really get some inspiration from such lectures and research work. So thank you very much, everybody, those who are participating in our webinar and our speakers. Thank you very much, sir. Thank you, Khan sir, for your work and your efforts. They are too great. So thank you very much on behalf of the organizing committee and on behalf of our Antarbharati Homeopathic Medical College. Thank you.
Thank you all of you. Thank you viewers. Thank you my speakers, beloved speaker, Dr. Nayak and Dr. Gore.
Thank you.
Thank you,
Namaskar,
Akhilesh sir.
namaste, namaste, and namaste, namaste.